Good morning, everyone. There we go. This week, we are starting a new chapter. We'll be covering cellular respiration today and Wednesday and Friday. And Friday will be the last day of material before our second exam, which will be next Monday, a week from today. So I hope that everyone has been keeping on top of the chapters we've been covering the past couple of weeks. Because we have an exam on Monday, Joshua will hold his additional office hour this Tuesday at 2, in addition to his regular Thursday morning office hour. I will have chat office hours today at 12:30, and then I have my Zoom office hour on Wednesday at 1 o'clock this week as usual. I'm going to direct your attention, those who have the e-text and the smart work animations. I would recommend that you look at the animations. There's a couple of different ones. Glycolysis, I'll show everybody a quick snippet of that one. They're pretty long. They're 7 to 8 minutes long, and they go through the entire process of glycolysis, which we will be doing today in class. And they go through the citric acid cycle. And then there's also animations of the electron transport chain and ATP synthase, which we will be talking about later this week. So I'll remind you to take advantage of those animations, really nice additional way of looking through the processes that we're covering in this chapter. So I hope everyone has had a chance to find and log into your Reef account. Make sure that you have joined our course. So you're ready to answer clicker questions, because I've got a clicker question right away. I'm going to ask you sort of a review question right away before we even get started. And our first part of cell respiration today, we'll be going through the different steps of glycolysis. So starting with our first slide, introducing our chapter, cellular respiration, the process by which cells can extract the energy from food molecules, from the fuel that we take into our cells. How does any living cell extract energy and transfer it to the mobile energy carrier, ATP? And what this figure is showing is just a part of cellular respiration, a part of aerobic respiration, that eukaryotic cells with their mitochondria will be able to use the products of glycolysis. We're going to talk about glycolysis that occurs in the cytoplasm of all cells, but the most efficient form of cellular respiration, in the presence of oxygen, will be able to further oxidize the products of glycolysis, extract more energy, and be able to generate a huge amount of ATP. So we'll be talking about these parts, the stages of respiration that occur in the mitochondria. We'll start looking at those on Wednesday and continue that on Friday. So for today, we're going to start by looking at glycolysis, this ancient step or stage, ancient process that is present in all cells, because all of these steps occur in the cytoplasm. And so the first question then is, we discussed this last week. Why does the cell go through all of these steps of glycolysis first, and then can continue in the following stages of cellular respiration? Gradual oxidation of glucose, that sugar molecule, as opposed to glucose, a sugar, undergoing combustion, a single step burning of sugar to produce carbon dioxide and water. Because either way, whether it's in the cell, in a living cell, or occurs outside of the cell, glucose will be oxidized to carbon dioxide in the presence of oxygen, which is reduced to H2O. Let's get our votes in, about 5 more seconds. What's the advantage of the cell evolving all of the enzymes, catalyzing all of the different steps of this long biochemical pathway of gradually oxidizing glucose? All right, let me close it up. And the advantage is extracting that energy, those high energy electrons carried in the glucose molecule, extracting them, and even especially if we continue to completely oxidize all of the carbons in glucose, extracting in usable amounts. Now, this cellular respiration will release the same amount of energy from glucose, whether it is gradual oxidation or one quick oxidation. Actually, much of the energy is converted to thermal energy, but our cells use that lower energy, lower form of energy, the more disordered form of energy, thermal energy, to keep ourselves warm or to overcome the activation energy required for some types of, well, actually for all chemical reactions. And the same amount of carbon dioxide is produced in either type of oxidation, a quick oxidation in vitro or through cell respiration. And so let's look at the steps. The first part of oxidizing glucose, the very first part, is glycolysis. The 10 steps of glycolysis, this most ancient biochemical pathway, 10 enzymatically catalyzed steps that we can actually divide into 3 different stages. We start with a molecule of glucose, a 6-carbon carbohydrate. And the first phase, the first 3 steps of glycolysis requires an investment of energy in that 2 molecules of ATP will be used or consumed in this biochemical pathway. The second stage is the cleavage of a 6-carbon sugar molecule broken down into 2 3-carbon sugars. And then each of those 3-carbon sugars are oxidized. And in that stage, 4 ATPs are generated. We end up at the very end of glycolysis with 2 molecules of pyruvate, 2 molecules of the 3-carbon pyruvate. So we're going to be looking at each of those steps, which again, we can summarize, and this is how I want you to be thinking about glycolysis. We'll be looking at each of the enzyme catalyzed steps. I will show you the substrate, I will show you the product of each step. The enzyme is listed, but what I really want you to know is what are the substrates and products. I don't expect you to memorize their structures, but if you follow the carbons and if you remember, and what I do want you to remember is the chemical change that's occurring, then you should be able to follow the all of the steps of glycolysis. So again, we're starting in our first stage. We have 1 6-carbon glucose. The initial energy investment stage requires the hydrolysis of ATP. So we're going to use 2 ATPs to be able to form fructose 1,6-bisphosphate, still 6-carbon, which will be cleaved into 2 molecules of glyceraldehyde 3-phosphate, a 3-carbon sugar. Then we enter the energy generation phase, where that glyceraldehyde 3-phosphate will be oxidized, electrons will be removed and transferred to our electron carrier, NADH, which is reduced, and phosphorylation, meaning transfer of a phosphate from a substrate to ADP, will synthesize ATP molecules. And then we end up at the very end with 2 molecules of our 3-carbon pyruvate. So as we go through the steps, you don't need to memorize the molecular structure. You don't need to memorize the name of the enzyme, but I do want you to know what are the substrates and what are the products of each stage and what type of reaction is occurring. So let's start. Our first step, we start with our 6-carbon glucose. There is carbon 1, this is the ring form, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 of the ring, and then carbon 6 extends above the plane of this ring. Glucose is phosphorylated. Glucose is converted to glucose 6-phosphate. This phosphorylation event involves the hydrolysis of ATP. The enzyme that carries out this reaction is called hexokinase. So what we need to really remember here is this reaction is energetically favored in the forward direction because it's driven by the hydrolysis of ATP. Right? Hexokinase uses ATP, moves a phosphate from ATP and moves it to carbon 6. So now we have a phosphorylated sugar. We have a phosphorylated glucose that we call glucose 6-phosphate because it's carbon 6 that is now has been phosphorylated. Right, that's step 1. The other important feature of this first step, remember how glucose can diffuse across the membrane, right? Glucose can bind to a glucose carrier protein and be transported across the membrane, but it can be transported in either direction, down its concentration gradient, from outside to inside, or even inside to outside. But once glucose has been phosphorylated, if we remember, a phosphate group has 2 negative charges on it. And so now this glucose 6-phosphate is negatively charged, which makes it impossible for it to diffuse back out of the cell again. So this first step actually traps the glucose inside the cell. And then this, as we'll see, this glucose 6-phosphate is a substrate for the next step of this biochemical pathway, glycolysis. All right, so we phosphorylate the glucose first, and then step 2 is that glucose 6-phosphate undergoes an isomerization to fructose 6-phosphate. So this enzyme right here, phosphoglucose isomerase, will convert the glucose 6-phosphate from its ring form to its linear form, what we call the open chain form. And we've just numbered all of our carbons again, just to remind us and make it easy to follow what's happening in this step, where the this linear form of glucose shows us that glucose is an aldehyde sugar, right? There's our carbonyl at the end of the chain. And this aldehyde sugar will be converted to a ketose sugar because this enzyme will just rearrange some of the, oops, sorry, don't need to be going ahead. We'll just rearrange some of the covalent bonds such that the carbonyl will be at carbon 2. So it's converting an aldehyde sugar to a ketose sugar. And then this linear form of this 6-carbon sugar will be converted to its ring form. Again, this enzyme is doing all of these steps. And the ring form will then form the ring in a slightly different way. So now we have a 5-sided ring instead of a 6-sided ring. We still have 6 carbons, but now 2 of the carbons are extending above the plane. So carbon 1 is extended above the plane of the ring, as well as carbon 6 above the ring. So this isomer didn't require any energy for this conversion to occur. glucose 6-phosphate has been converted to fructose 6-phosphate. Now, in the next step, we'll see fructose 6-phosphate is the next substrate, and it will be phosphorylated again. So here we have our fructose 6-phosphate, carbon 6 is phosphorylated. The enzyme phosphofructose kinase will transfer, as a kinase does, right? It transfers a phosphate from an ATP to a substrate. And therefore, this ATP is consumed, ATP hydrolysis occurs. That is the spontaneous reaction that's driving this reaction forward, right? From left to right. That phosphate from the ATP will be transferred to carbon 6. So we end up with a molecule, fructose 1,6-bisphosphate, meaning 2 phosphates on this sugar. ATP has been converted. ATP has been converted to ADP, and we now we've consumed that ATP and we've phosphorylated. So now both carbon 1 and carbon 6 are phosphorylated. This is the energy consumption phase. 2 ATPs have been used in these 3 steps to generate fructose 1,6-bisphosphate. The next step is the next phase. This is the splitting step. This is the step that gives glycolysis its name. Glycolysis means sweet splitting, the splitting of the sugar. Our 6 carbon, there's carbon 1, 2, 3, 4, 5, 6, our 6 carbon sugar, ring form converted to its open chain form, will be cleaved into 2 smaller sugars, cleaved right at this bond here by aldolase. Aldolase produces 2 3-carbon sugars. There's 3 carbon, this acetone, dihydroxyacetone phosphate, and there's carbon 4, 5, and 6 make up our second 3-carbon sugar. Both of these are phosphorylated triose sugars, the smallest type of sugar that exists, a 3-carbon sugar. One of them is a ketone and the other is an aldehyde. And each of them carries 1 phosphate at the end of their carbon chain. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Right? So that's our middle, the second phase of glycolysis is the cleavage phase. Now, what's really important for glycolysis to be able to overall generate more ATP than was used to get to this state is that in the next phase, or the next step, the next step of glycolysis, is that dihydroxyacetone can be converted to another molecule of glyceraldehyde 3-phosphate. So this is an isomerization step. One of these 3-carbon sugar can be converted to the other. But what we're going to see is that glyceraldehyde 3-phosphate is the substrate for the next step. And so as long as glyceraldehyde continuously is converted to the next step of glycolysis, then dihydroxyacetone is continuously converted to another molecule of glyceraldehyde. And so what we end up really is for every molecule of glucose, we end up with 2 glyceraldehyde 3-phosphates. Okay, triose is a sugar. Okay, I'm not sure if I'm supposed to be paying attention to what's going on in the group chat here. We've got a lot of conversation about the biochemistry, which is great, and a lot of people have probably learned this in biochemistry class, but I'm trying to highlight number 1, following the carbons, following what's driving this reaction forward in step 1 and step 3. the hydrolysis of ATP is driving this reaction in the forward direction, and also allowing us to follow how this molecule is getting smaller and where the oxidation step occurs, which is actually our very next step. So with one molecule of glucose, we end up by this step, by step 5, we end up with 2 3-carbon sugars, but really we end up with 2 molecules of glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is the only substrate that is oxidized in glycolysis. So our next step is really, again, they're all important steps, but our next step is the oxidation step of glycolysis, the only step where a carbon is oxidized. And so what we see here is this enzyme, glyceraldehyde 3-phosphate dehydrogenase, and again, the enzymes are listed. The enzymes are in some ways descriptive of their, they were named by the scientists who discovered them, descriptive of what they're catalyzing. This dehydrogenase is removing hydrogen atom. And this actually carries out 2 sequential reactions, which we're just summarizing here. Right? It's much more complicated than this overall reaction that we're showing. But our glyceraldehyde 3-phosphate is oxidized. It's going to lose this hydrogen atom. This hydrogen atom is going to be transferred, and really what's important is the electron of this hydrogen atom is going to be transferred to the electron carrier NAD+. So this step, the carbon is losing electrons, they're being transferred to electron carrier, and we produce NADH. In the process, that oxidation, and remember, oxidations release energy. The energy that was released by this loss of electrons of this hydrogen atom drives the addition of this inorganic phosphate such that it can be transferred to that carbon. So 2 steps. Loss of hydrogen atoms and the addition of a second phosphate to this 3-carbon molecule. So now we have another bis, as in 2, we have a 1,3 bis phosphoglycerate, a glycerate that has 2 carbons that have phosphate. Now this phosphate added to this oxygen here, this is an anhydride bond. This is a high energy bond. There's high energy intermediate, high free energy in the electrons that are joining this phosphate to this oxygen to this carbon. So this is a high energy molecule. The reason this is a high free energy molecule is because the energy that was released by removal, the enzyme removing the electrons, allowed for the formation of this high energy bond. We don't need to know what the free energy change of hydrolysis of these are, but suffice it to say that this is a high energy molecule. So, and remember, we've got 2 of these, right? Because this reaction occurs twice for every molecule of glucose, because that previous step had actually generated 2 glyceraldehyde 3-phosphates. So we've actually generated 2 molecules of NADH. And now we're going to see what happens to this high energy bond in this 1,3 bisphosphoglycerate. Now we're going to start generating some ATP. We generate ATP because our, let's see, I'm on the wrong page here. What we're going to see is this kinase, phosphoglycerate kinase, will transfer this phosphate from this high energy anhydride bond and transfer it to ADP to form a different anhydride bond of ATP. The free energy change from this bond to this bond is greater than the free energy change required to convert ADP to ATP, which allows this reaction to occur. So we're transferring a phosphate from this substrate, 1,3-bisphosphoglycerate, to ADP, which is what we call a substrate level phosphorylation, which results in the generation of ATP. And again, this happened twice. So now we've already recouped the 2 molecules of ATP that were consumed in the earlier steps of glycolysis. And we end up with a molecule that we call 3-phosphoglycerate. Right? Only carbon 3 remains phosphorylated. The next step, step 8, is an isomerization, and this enzyme will transfer the phosphate from carbon 3 to carbon 2. So what it does overall is it converts 3-phosphoglycerate to 2-phosphoglycerate. Now this phosphate is in a position that this molecule is now the substrate of step 9, in which a dehydration event, removal of this hydrogen and that hydroxyl, removal as a molecule of water, causes a change in the covalent bonding of the remaining atoms that forms what we call an enol phosphate linkage. And again, this is now a very high free energy, essentially a very highly unstable bond between this carbon and this oxygen and this phosphate. We call this phosphoenol pyruvate. This high energy molecule will be our last substrate for our last enzyme, step 10, where phosphoenol pyruvate, sometimes called PEP, will transfer its phosphate to ADP. Pyruvate kinase will catalyze that reaction, transferring a phosphate. Again, the change in free energy from phosphoenol pyruvate to pyruvate, the loss of that phosphate is a greater change in free energy than the energy that's required to transfer that phosphate to ADP to synthesize ATP. So it's driving the synthesis of ATP. Removing this phosphate is driving the synthesis of the addition of the phosphate to ATP. Again, making this energetically favorable in the forward direction. And we end up with our molecule of pyruvate, 3-carbon molecule of our pyruvate. We have 2 of them because for every molecule of glucose, these last 5 steps occurred twice. So we end up with 2 pyruvates. So what happened in summary? 1 6-carbon sugar has been converted to derivatives that required 2 molecules of ATP, which was then split into 2 smaller 3-carbon sugars, each of which underwent the next 5 steps. The first one was oxidized. The glyceraldehyde 3-phosphate was oxidized, transferring the high energy electrons to NADH, and the subsequent steps used the transfer of phosphates from high energy, very unstable bonds to ADP to generate 2 molecules of ATP twice. So we used 2 ATPs to start, we synthesized 4 ATPs, we end up with a net of 2 ATPs and 2 NADHs. This multiple enzyme 10-step process converting glucose to pyruvate, but the reason, the goal is to generate ATP. And so many organisms will use glycolysis as a way to generate just a little bit of ATP over and over again. Now, one thing we need to keep in mind, of course, is that if this pyruvate builds up in the cell, all of those enzymatic reactions can catalyze, or the enzymes can catalyze reactions in either direction. So if pyruvate is building up in the cell, glycolysis will stop. So a cell that is undergoing aerobic respiration will use our pyruvate. Our pyruvate will be fed into another round of enzymatically catalyzed reactions in order to remove pyruvate and further oxidize all those carbons. So aerobic respiration will continue to oxidize pyruvate, but some cells don't have access to oxygen. And we'll see a little bit later what happens to the pyruvate to make sure that the cell is constantly removing the pyruvate, converting it to something else so that glycolysis can keep continuing. So what I'd like to do here is show us that we have, as one example of the animations that are available, they have a very nice description. It's 7 minutes long, so I don't want to show it here, of all of the different steps, the overview, as well as each of the individual steps of glycolysis, watching all of the atoms move around. But I think what I can do here is, my plan was to stop this for a second. I should have it ready here. And we can watch sort of the first minute to let you know what this is going to be like. Okay, so I don't want to go through all of that here, but if you're interested, you can see what how they're actually showing animating all of the different chemical changes that are occurring, which I think is really nice. So for our purposes, we went through each of the individual steps. I told you you don't need to memorize the structures of each of the steps. You don't need to memorize the enzymes of each of the steps, but by looking and following them and maybe even drawing them for yourself, you will know. What I want you to know is what is the substrate, what's the product of each step, and what has occurred at each step. What is the chemical change that's occurring? And this is the kind of question I want you to be able to answer. Whoops, I'm going the wrong direction, sorry. There we go. All right, so for example, you should be able to tell me which of these reactions, which of these steps of glycolysis is an isomerization. And then I'll ask you what are the others? The other ones that are not an isomerization, I'm going to ask you to tell me what kind of chemical reaction are each of those. Has everybody had a chance to vote? Let me close it up. All right, closing up. And the only isomerization that's illustrated here would be B. Can't select it, I'll do that later. And B is, there we go, glucose 6-phosphate converted to fructose 6-phosphate. That's an isomerization. What would we call A? Glucose converted to glucose 6-phosphate is what kind of a reaction? That's a phosphorylation. What about fructose 6-phosphate to fructose 1,6 bisphosphate? That's a phosphorylation as well. And glucose to glucose 1-phosphate is also a phosphorylation. Which one of these steps is not part of glycolysis? Exactly. Glucose is only phosphorylated to glucose 6-phosphate, right? And then isomerized. So this step is not part of glycolysis, right? That's part of a different chemical reaction or different biochemical pathway. So again, a cell, and again, every cell, right, carries out these 10 steps of glycolysis, the most ancient biochemical pathway. This occurs in the cytoplasm. But some cells have to get rid of their pyruvate in a different way if they can't further oxidize it, if they are under anaerobic conditions. So in the absence of oxygen, that pyruvate, as well as the NADH, have to be taken out of the cell or consumed or changed, and that occurs in the process of fermentation. Remove the pyruvate, reduce it. If it can't be oxidized anymore, the cell needs to reduce it to get rid of it. And the cell needs to continually regenerate NAD+ to be able to extract more electrons from glyceraldehyde 3-phosphate at this step. So that's what we're going to look at next. Fermentation is the process by which glycolysis occurs, but the pyruvate that is being formed is reduced. And it's reduced to a molecule that can easily exit the cell so that glycolysis can keep moving forward. In muscle cells, when muscle cells have used up all of the oxygen and they need to keep making a little bit of ATP, some bacteria that are used for producing yogurt, some other types of fungi that are used for making cheeses, that pyruvate is converted to lactic acid. It's reduced to lactic acid. And along the way, the NADH is oxidized to regenerate NAD+. In yeast, used in brewing or baking, as well as in some bacteria, anaerobic conditions, those pyruvates are converted first to acetaldehyde, and the pyruvate loses a fully oxidized carbon in the process, and then the acetaldehyde is reduced to ethanol. That reduction, again, allows for the regeneration of the oxidized form of NAD+. As long as fermentation can continue, the removal of the pyruvate and the regeneration of NAD+ electron acceptor, glycolysis can keep going, and every molecule of glucose generate a little bit of ATP. So let's look at each of those separately. First type of fermentation, this is what occurs in muscle cells. Once our muscle cells have used up all the oxygen available, glucose converted to pyruvate. The process of glycolysis yields a net to ATP. a net of 2 NADHs. The pyruvate will be reduced to lactate. Lactate's the ionized form of lactic acid. Right? That lactic acid can leave the cell. Lactic acid will leave the cell and will eventually find its way to the liver, where that lactic acid can then be converted back to pyruvate. So it can be further oxidized. But this process of reducing, and these are enzymatically catalyzed reactions too, the pyruvate reduced to lactate or lactic acid is coupled to the oxidation of NADH, regenerating the NAD+, which is necessary for the cell to be able to continue driving glycolysis. The other type of fermentation, this is the fermentation that occurs in yeast cells. Glycolysis produces, for every 1 molecule of glucose, 2 molecules of pyruvate. As we can see here, the pyruvate is converted to acetaldehyde. This carbon here will be lost as a fully oxidized carbon. That's where the carbon dioxide gas bubbles come from. The acetaldehyde is then reduced to ethanol. And again, the ethanol is a more soluble form of the pyruvate. Ethanol can leave the cell as well. That reduction of the acetaldehyde to ethanol drives the oxidation of the NADH, recycling or regenerating NAD+, so that glycolysis can keep continuing. So we're getting rid of excess pyruvate so we don't come to equilibrium, so that the cell does not, won't have glycolysis stop, and regenerating the electron carrier that allows that glucose to be oxidized. Right? Both the carbon dioxide can leave the cell, the ethanol can diffuse out of the cell, constantly removing the pyruvate so glycolysis can move forward. So what are we seeing here? These are some of the potential products. Which one listed here is the final product of fermentation in yeast? Now, again, I'm not going to ask you to memorize any of these structures for our test, but since we've just described them and you have your notes, see if you can tell me which is the final product of fermentation in yeast. Everybody had a chance to vote? 5 more seconds. I see a hand raised. You have a question? Erica has a hand raised. I see 2 hands. Anybody got a question? Okay. All right, let's close it up. Final product of fermentation in yeast is the ethyl alcohol, right? Ethanol, which would be B, right? So here is our 2-carbon ethyl alcohol. What is D right here? Anybody want to tell me? So fermentation of yeast produces carbon dioxide and ethanol, but right here, what is D? 3 carbon, 1 carbon is almost fully oxidized. It's going to easily be lost. We have 2 more carbons that can be oxidized. That's pyruvate, right? What we're going to see is in the presence of oxygen, this pyruvate can further be oxidized. But in the absence of oxygen, in yeast fermentation, the pyruvate is converted to, what's this here? C. Converted to, pyruvate's converted to, right? That carbon dioxide is removed, leaving us with a 2-carbon molecule, acetaldehyde. And then the acetaldehyde can be reduced to ethanol. Right? A way of getting rid of the pyruvate that's building up so that glycolysis does not come to equilibrium. We want that process to be spontaneous, overall spontaneous, keep moving forward. Right? Now, under aerobic conditions, the pyruvate, the carbons of pyruvate can be further oxidized. This happens in the mitochondria. So pyruvate has to be transported into the mitochondria. The electrons that are carried in NADH are also transferred into the mitochondria. Pyruvate has to be converted to a 2-carbon molecule we call acetyl-CoA so that it can enter the citric acid cycle, which will further oxidize it. And the electrons of NADH are used to power the electron transport chain. And that we'll be talking about later this week. So everything from now on is occurring in the mitochondria. Remember, our mitochondria has 2 membranes, an outer membrane and an inner membrane. The inner membrane has these extensive folds, adding more surface area for all of the enzymes that are required for electron transport, the oxidative complexes. And the membranes also separate 2 compartments. We have this compartment here, the innermost compartment we call the matrix. This is where citric acid cycle occurs. All of the enzymes driving the citric acid cycle, the enzymes that are converting pyruvate to acetyl-CoA, that pyruvate has to be transported across both membranes. All of the following reactions are occurring in the matrix, and having these 2 membranes also forms a second compartment that we call the intermembrane space. So we actually have 2 compartments separated by an inner membrane, and that's going to be really important for electron transport and generation of ATP. So everything that's going to happen next, again, in the presence of oxygen, is oxidation of pyruvate, further oxidation in the citric acid cycle, all occurring within the matrix of our mitochondria. And I really like this. This is where we're going to end today, and this is where we'll pick up on Wednesday. A nice figure of sort of the functional portions of our mitochondria. 2 membranes. Here's our outer membrane. Here's our inner membrane. We have our intermembrane space. The inner compartment we call the matrix. The pyruvate, as well as other molecules, other breakdown products from other fuel molecules like fatty acids, will be in 2-carbon products, will be transported across those membranes into the matrix. Pyruvate transported across the inner and outer membrane of the mitochondria into the interior compartment of the mitochondria. They can enter this cycle of catalyzed reactions we call the citric acid cycle, which will release more carbon dioxide that diffuses out of the mitochondria, then diffuses out of the cell, extracts more high energy electrons that are carried in carriers like NADH. Those electrons are then transferred to the electron transport chain, which resides within the inner mitochondrial membrane. And as we'll see later this week, transfer of those electrons drives the pumping of protons from the matrix side to the intermembrane space, creating a proton gradient that will drive the synthesis of ATP. So everything we're looking at here and what we'll be talking about the rest of the week is in the presence of oxygen. More electrons can be extracted from our pyruvate those electrons will be used to drive the synthesis of ATP. In the absence of oxygen, the pyruvate is reduced to molecules that can diffuse out of the cell and allow glycolysis to keep continuing. Just generating a little bit of ATP at a time, but as long as glycolysis can keep running, the cell can generate ATP that it needs to do the work of the cell, right? The other work of being alive. So we'll stop here. We'll pick up with our mitochondria and what's happening to our pyruvate. We'll pick up on Wednesday. All right, take a look at the summaries. Yes, what I really want you to be thinking about, what goes in, what goes out. If that makes sense, then all of the individual steps will be able to make sense as well. All right. If anyone has questions, I will be in my chat office hours at 12:30 today. Otherwise, I'll see you on Wednesday.